We are talking about chemicals of life. So we finished those chemicals of life, then we introduced nucleic acids, like how you've just reviewed our lesson, and we stopped on formation of a polynucleotide. And all videos are there, you can watch them over and over and over again. So today we are going to begin with the DNA structure and actually see what DNA is. Okay? And we shall try to talk about the structure that is acceptable right now. And it is the Watson-Crick model of DNA. That's what we shall talk about today. You know, science is developed gradually. And if you are to read about this story of DNA, it is really interesting. It is really interesting. But for the benefit of time, we shall not go into stories. We shall just go straight to the structure of DNA. Now, the Watson and Crick, these were two guys. Actually, one was a professor and the other was a student. One was a doctor and the other one was a student. Okay? I think, yeah, they are here. This was a doctor and the other one was a student. And these guys devoted their time to studying the structure of DNA, to identify what exactly DNA was about. What was that DNA and how was it able to store all the genetic information? Okay? All the genetic information. And so these guys, they studied and came up with a structure of DNA which tried to explain all this. And but up to now, we still believe in their structure. It was really good. It was really good. Now these guys, they first studied. First of all, there was a concern to identify. Yes, by that time, they knew that there was organic basis. This DNA was made up of organic bases. But we didn't know actually how these bases paired with each other. That some people agreed that purines, they pair with purines, and pyrimidines pair with pyrimidines. But when these guys studied the ratio of adenine and thiamine and that of guanine and cytosine in the DNA, they found out that the ratio was always constant. Okay? The ratio, the amount of thiamine and adenine were almost the same. The percentage was almost the same. And the ratio of guanine to cytosine, the percentages, they were also almost the same. And so these guys probably hypothesized that probably, yes, they believed there was pairing. So because of this, they probably believed that thiamine paired with adenine and guanine paired with cytosine. And so this was a great breakthrough, okay, to studying the structure of DNA. Because there was a confusion, okay? There was a confusion that, yes, DNA is made up of organic bases. But these organic bases, how are they paired? How do they do that? And because of that, they were able to convince us. Because if you see, the purines and the pyrimidines, purines have a double ring or double ringed structure, whereas pyrimidines have a single ringed structure. Now, if adenines paired with adenines, if purines paired with purines, and adenine paired with adenine, pyrimidine paired with pyrimidines, then the distance wouldn't be the same. Because if you have this double ring structure and another double ring structure pairing with a double ring structure, okay, and then here you have a single ring pairing with a single ring, it would create confusion. This distance cannot be the same. But when they studied the structure of DNA, they showed that from one strand to another strand, this distance was always constant throughout the entire DNA. So because of this, they were able to support their base pairing rule. Okay? So therefore, this showed that this is according to Watson and Crick. These guys hypothesized that the DNA consists of two parallel strands. In fact, if you want to be exact, then you can say two antiparallel, because these strands, they run in opposite direction. So if this one runs in this direction, then the other one runs in the opposite direction. But the two strands are parallel to each other, both having organic bases at right angles that point inwards, okay, and that these organic bases would pair up to form strong hydrogen bonds that held these two parallel strands close together. And so these guys came up with a base pairing rule, and they were like base adenine, which is a purine, pairs with base thiamine, which is a pyrimidine, and base cytosine, which is a pyrimidine, pairs with base guanine, which is a purine. And up to now, we still accept this base pairing rule. And the two bases form what we call the hydrogen bonds. And this is the structure that they hypothesized. Okay? You can see you have, after forming your polynucleotide strand, the two polynucleotide strands, they will lie parallel to each other to form that ladder shaped. Okay? And then because these organic bases, they orient inwards at right angles, they will form the intermolecular forces of attraction, which are the hydrogen bonds, like how you can see. And now there are these intermolecular forces of attraction that will hold these two strands parallel to each other. Okay? Yes. Now, that is the basic. Now that was internal. Now, they hypothesized that it is the structure of DNA was strongly wound or strongly twisted to form that helical structure, that helix, and that the strands would run in opposite direction. So if you have a look at this, okay, the two strands, yes, they are parallel to each other, but the two strands are not as straight as you may think a ladder is, or as a ladder, but rather they are strongly wound, they are strongly twisted, okay, to form that or to give that helical structure. And the two strands run in opposite direction, whereby if you have one strand that runs in this direction, maybe this is the 3 and this is the 5 prime, 3 prime, and where is the 3? The 3 end. If we draw this pentose sugar, where this is the O, and then you have carbon number 1, carbon 2, carbon 3, carbon 4, and this is the carbon 5, and you have the C2 OH here, and this you have the OH here. So this is carbon 1, carbon 2, carbon 3, carbon 4, and carbon number 5. So this one is going to be the 3 carbon or the 3 prime end, and then this one is going to be the 5 prime end for this strand. Then the next strand must run in the opposite direction, whereby this one will be the 3 prime end and this one will be the 5 prime end. For them to be able to pair up, for them to be able to form hydrogen bonds, okay, with each other, to form hydrogen bonds with each other. And this was key, okay, this was key as far as the structure and function of DNA is concerned. Because think about this, why should you have that? Why should you be double stranded? And why should these organic bases point inwards? And why should they form those strong hydrogen bonds internally, not outside? Think about that. Why? And relate to the role, to the function of DNA. It's a genetic material, so it should be stable enough. Imagine if these organic bases are just oriented outside. Then from nowhere, they would be easily distorted. These organic bases would be easily distorted. But because they oriented inwards, then they are strong enough. They are stable. Moreover, with a high molecular mass, so DNA is stable enough, okay, to become a nucleic acid, to become a store for genetic information. Then these strands aren't parallel, that's what we have said already, that run from the 3' to 5' direction, and the other one therefore must run from the 5 to 3'. They are twisted to form a double helix, like how you can see, to form a double helix. Why double helix? Now, I don't know if you've ever seen, okay, if you know these radios which use battery, you know shock absorbers of a car, these radios have some helix, okay, where the negative terminal attaches and then the positive terminal attaches to be able to use the dry cells. So now that is the helix, but for it, it is just one coil. But if you get the two helices and one runs in that direction, and then you get another one and it also runs in that very direction like that, then you form an identical. This what you form is identical to the structure of DNA. Even when we were young, we used to have some key holders. I don't know where those key holders went. They were like this. It was a long thread with some key hole this side and a hole this side where you would attach your key. I used to like that thing. I used to like it so much. So when you get the two of them and you bind them, so they form a structure that is identical to this DNA. So that's how these guys hypothesized their DNA to be. That it's a double helix that is highly twisted or that is highly wound. Okay? Or that is highly, yes, wound. That word is good. Wound to form a double helix. Then, since the organic bases point inwards at right angles, okay, then these organic bases are able to form intermolecular forces of attraction between themselves. Okay, these ones, this is what I'm talking about. Now these intermolecular forces of attraction between themselves, they end up forming those, it forms a ladder-like structure, okay, with organic bases as those, they are called, they are rungs, the rungs in a ladder. If you have your ladder like this, then these, should I say this, whatever that run through, they are called rungs. Then these are pillars or posts, whatever. So now these organic bases, they form something like a rung within this ladder. And why? So that you form that stable structure of DNA, okay, that is going to hold your DNA molecules close together. Now if you try to identify the DNA is stable in a way that the distances between the two phosphate backbones or the two backbones from here up to here, it will always be constant from DNA to DNA. From this side, even if you get another twist, if I twist like this, so, and this is my DNA, so the distance from this to this, it will always be constant with another distance from here to here, okay, and even the distance from here to here, it is also constant, okay, which makes DNA to be stable and hence becomes a genetic material. Because man, storing information, storing this information, it is key. It requires enough care. Even we are going to see in replication, we are going to talk about DNA replication. We shall see that there is a lot of carefulness, so that you copy well your DNA. Then the base pairing rule, like we said, the base adenine, which is a purine, it will pair up with base thiamine, which is a pyrimidine, and the base guanine, which is a purine, it will pair up with base cytosine, which is a pyrimidine. And the two are held by stronger hydrogen bonds that will keep that stable structure of DNA. Now, after forming all this, then you won't store your DNA in the cells as this. You know, if this is a cell, then you expect to find your DNA in the nucleus. Now, after forming this helical structure of DNA, within the nucleus you have there protein molecules called histone proteins. Histone proteins. Now it is this DNA that is going to coil itself around these histone proteins, so that you form certain structures. This is a histone, the DNA together with a histone protein, and then you form another one, and then you form another one, and then you form another one. If you still remember the others, the peak that I used on my very first slide, maybe I can take you there. Yes, this is the peak I was meaning. So after forming such a helical structure, now this helical structure must be condensed to form chromosomes, and the chromosomes can easily be stored within the nucleus. Actually, they are the ones which are even replicated. They are the ones which can be, by cell division, you transfer them to different cells. So it is chromosome. So that's why anything that occurs on the chromosome, it will result into a change in the DNA. So therefore, we are going to coil our DNA, our helical structure, onto what we call the histone proteins. Okay? The histone proteins. And after coiling, even these histone proteins, These coiled DNA together with some proteins, they also undergo further coiling, and you find you have the coiled protein molecules. So that in the end, you will end up coiling your DNA to form what we call a chromosome, and it is stored constantly within a cell. Okay? Within a cell. So that is some basic understanding of what DNA is in your cell. Any queries for me? Feel free to ask.